Our next speaker is Scott Friedman, who's from Mount Sinai School of Medicine, and he's going to tell us about From Fibrosis to Cancer and Back, the KLF6 tumor suppressor gene story.
Thank you, Arnie. It's really a delight and a privilege to be here. It's a different kind of audience. I'm not a virologist, and so as you'll sense, I frame the approach to our problem a little differently. In fact, one of my main day jobs is as a liver doctor, so hopefully you'll get a sense of how we attack the problem. So for many years, we've been interested in the liver's response to chronic injury, and more specifically, the fibrotic response. And the way we've envisioned this over now 25 years is that effectively what the liver is doing in response to chronic injuries is mounting a wound healing response. The main diseases that lead to fibrosis that we see clinically, and most important for this audience, are hepatitis B and C, but increasingly non-alcoholic steatohepatitis associated with obesity, as well as alcohol abuse. And to complete the story, there are other causes of liver injury and fibrosis as well, but we've focused primarily again on this problem because it still represents the largest fraction of our patients with progressive liver disease and ultimately with hepatocellular carcinoma. So there's one thing that's very instructive to think about in managing patients with liver disease, which is quite distinct from most other chronic inflammatory diseases, and that probably reflects the liver's unique regenerative capacity. So that whereas many fibrotic diseases have a natural history of anywhere from 2 in the case of lung disease to 10 years in the case of renal disease, for most patients with chronic liver disease, they have a smoldering hepatitis that can vary in progression rate, but generally is between 10 and 50 years. And ultimately, that culminates in the end-stage scarring state that's known clinically as cirrhosis. Now, one of the most interesting and still perplexing aspects of this disease state is the fact that particularly in hepatitis C, a fibrotic liver is virtually always required for hepatocellular carcinoma to emerge. And I don't have an answer here, but this currently is really one of the most intriguing questions for me and something we're beginning to attack experimentally. That is, what is so permissive about the fibrotic milieu that allows cancers to emerge, recognizing that HCV as an RNA virus is not integrating? In contrast to hepatitis B, where others in the audience, particularly Don Ganem, can tell us and me a lot more about the pathogenesis or the viral oncogenesis of HBV. And in this disease, in contrast to hep C, of course, tumors can emerge in non-cirrhotic livers, although the cancer risk increases in HBV just as it does in HCV as fibrosis accrues. Fundamentally, however, hepatocellular carcinoma represents a major problem and one that we're really only now beginning to make therapeutic as well as diagnostic inroads. And certainly in major liver centers such as ours at Mount Sinai, liver transplantation represents virtually the only curative therapy for chronic liver disease. And just to sober you up a little more, hep C always recurs when we do transplant, and it's become a major problem now that transplantation has been well established, that many patients with hep C come back to us 5 and 10 years later with cirrhosis again, requiring another transplantation. And certainly what you can imagine with the heroic nature of liver transplantation, this is by no means the cure-all for the hundreds of millions of patients or more worldwide with chronic liver disease. And so obviously we need to understand a little bit more the sequence of events, particularly the molecular pathogenesis of fibrosis, as well as the molecular pathogenesis of carcinoma that emerges from this fibrotic milieu. Just to underscore the importance of hepatocellular carcinoma, this graph illustrates the trends in U.S. cancer mortality rates, emphasizing that among all cancers, which overall generally are decreasing in the annual incidence, liver cancer represents the fastest rising cause of cancer mortality in the Western world. It's a huge problem. In our institution last year, we evaluated over 450 patients with hepatocellular carcinoma, and we recognize that this is probably just the tip of the iceberg. So what do we know about carcinoma risk in the 2 major viral infections that we see? Well, in hepatitis C, the annual risk is 1 to 4%, but this is only once cirrhosis has developed, as I've already emphasized. It seems to be highest in patients who acquire their infection from blood products, presumably, or although actually not really known, because of the dose of viral inoculum. Hepatitis B-related hepatocellular carcinoma is directly related to the duration of infection and viral replication. Cirrhosis is also a risk factor, and what we've learned recently from clinical trials is that sustained suppression of viral replication, lower DNA or decreased or absent DNA levels leads to markedly decreased rates of hepatocellular carcinoma in non-cirrhotic patients. So there's a good case for major advances in antiviral therapies for Hep B, not to mention the extraordinary outcomes of nationwide vaccination programs such as those in Taiwan. So with that backdrop, I'm going to frame my comments in the next few minutes around three general areas. One is to review for you briefly the cellular mechanisms of fibrosis, really in the context of how they may create a milieu that favors carcinoma emergence. Secondly, and this is largely for Arnie because he's been involved as an advisor in some of this work through a company that I've been working with, that is, what are the genetic determinants that indicate or that drive a patient to develop scar quickly or slowly? And then finally, I'm going to focus in on one particular mechanism that we've been studying for about 10 years now, that of the KLF6 tumor suppressor, and also to illustrate how it's yielded a number of surprises that we think are quite informative in cancer pathogenesis. So let me start with a very simple paradigm, but one that has really held very true for us in our experimental models for many years. Several years ago, we became interested in what the source or sources of scar are in that chronically inflamed liver, and we focused on a cell type shown in this drawing known as the hepatic stellate cell. It's the major cell source or cell type storing vitamin A in the body, and that's illustrated by these lipid droplets surrounding the nucleus. And at least histologic studies suggested for decades that as the liver became injured and fibrotic, these cells adopted a more fibroblast-like morphology. And it was really based on the vitamin A content that we established a method for isolating these cells in pure primary culture from rat and then from humans, and that really opened the door for us to really address their potential role in fibrogenesis. And simply put, what we understand now is that this cell type undergoes what we've called an activation response, which really represents a transition to a cell that is fibroblast-like, associated with a lot more scar, and that in turn leads to deleterious effects of surrounding cell function, something I'll illustrate a little more dynamically if this works in the video here. So what you're looking at here is the same organization of the sinusoid. These are the endothelium that are fenestrated or have pores, hepatocytes, and this is the subendothelial space. Now, if we focus in, as the liver becomes injured, this hepatic stellate cell begins to replicate or multiply, and it becomes a more contractile cell type known as a myofibroblast, losing its vitamin A droplets and elaborating a large amount of scar as well as a number of soluble molecules that mediate the regulation of scar deposition and degradation, TIMPs, metalloproteinases, and also hyaluronic acid. So the net outcome of the accumulation of all this scar is, as I said, the loss of differentiated function. So I want you to focus on these microvilli, which really are intended to indicate the differentiated state of the epithelial compartment or hepatocytes in the liver. And as you can see, they lose, this is of course a diagram, but it's intended to illustrate something that we know clinically is quite important, which is as the liver fails, as scar accumulates, one of the hallmark features clinically is that there's a loss of differentiated function of the organ, and that's intended to represent, or this diagram is intended to represent why hepatocytes may lose their differentiated function, why they no longer can metabolize drugs, why they can no longer generate clotting factors and clear substrates as well as they do in the healthy liver. So from that simplistic paradigm of stellate cell activation, those of us in the field have really been able to piece together a far more comprehensive and integrated view of how these stellate cells respond to injury, and that's illustrated in this slide, in which an initial injury response, presumably from oxidative stress, from apoptotic bodies, from lipopolysaccharide, and other paracrine stimuli, some of which may be virus-specific, yield a series of pathways that, if you will, represent a conspiracy to make more scar. And I'm not going to go through each of these, but there is a series of pathways that are each driven by their own cluster of cytokines and cytokine receptors, all of which, again, generate increased scar. One other important point is the emergence of stellate cells as a central player in the hepatic inflammatory and immune response. These are not just passive targets of inflammatory mediators, but rather they're in their own way able to modify the inflammatory milieu in ways that I don't have time to review. Perhaps more interestingly is, if we're going to therapeutically alter this pathway, how do we get rid of these activated cells? Well, it turns out nature does that very well, and most of the evidence to date suggests that they do so through programmed cell death and/or senescence. So a very beautiful paper by Scott Lowe and his group in the last year underscored the capacity of these cells to undergo p53-driven senescence and ultimately yielding an apoptotic cell associated with a number of changes that would be predictable in regulating cell death. And so this really is a framework that has yielded not only a better insight into pathogenesis of fibrosis, but from a therapeutic potential, every one of these molecules is being targeted in one way or another in the pharmaceutical or biotechnology space. And so we're really on the cusp now, we think, of developing effective antifibrotic therapies built around this and related models. So from the clinical perspective, I alluded to the fact that when patients have hepatitis C, they have a remarkably variable progression of scar. On average, when a patient is infected with hepatitis C, he or she will take 30 years to span the histologic spectrum from a normal liver up to a cirrhotic liver. And this is one of the major staging systems we use to quantify scar in a liver biopsy, F4 representing that cirrhosis state. So again, on average, 30 years to progress. And yet there's variability that means that some patients who are infected within 10 years become cirrhotic, and other patients over the course of 40 or 50 years have still not progressed to a state where they have any real clinical complications. And I need to underscore that probably up until METAVIR stage 3, these patients are completely asymptomatic. Many of them don't even know they have hepatitis C. So what accounts for that remarkable progression in scar formation in patients with hepatitis C? Well, interestingly, it has nothing to do with either the amount of virus or the genotype of that virus. There are some external features or factors that one would predict and in fact do contribute. Males progress more rapidly than females, particularly premenopausal females. Patients who drink alcohol to excess and other risks also contribute, but they don't really explain most of the differences in this variability. And so that's raised the question that's been addressed by Celera Diagnostics of what is the genetic contribution to fibrosis progression. And so in the next couple of slides, I just want to touch on how that's been attacked experimentally using clinical samples in a genome-wide association study, yielding data that you see here that was published last year. And this is a so-called cirrhosis risk score in which Celera, and the team was led by John Sininsky and Hongjin Wang, did this broad survey of patients with a defined progression rate of fibrosis and compared, of course, those who progressed slowly and rapidly. And what emerged from this very stringent approach was a series of 7 SNPs that in aggregate create a spectrum of risk, so that patients who have none of the risk SNPs progress slowly, patients that have all 7 of the risk SNPs progress very rapidly, and there's a number that goes in between that's a continuum that predicts risk. The problem, as many of you are aware with genome-wide association studies, is there's often no biologic link to the disease. It's simply an association. And so Celera really approached us as folks who are working in the fibrosis area to ask the question, or for us to answer the question, how could some of these SNPs actually alter the biology? Now, we worked through a number of these, but I'm just going to focus on one for the interest of time today, and one that's probably familiar to many of you, Toll-like receptor 4. So, as you probably know, Toll-like receptors are a family of mammalian transmembrane pattern recognition receptors. They each have their own repertoire of ligands. In the case of TLR4, the endogenous ligand, or the exogenous ligand is LPS, but there are additionally endogenous ligands. But what was interesting is that from the genome-wide association study, or the gene-centric association study, 2 of the SNPs that emerged were associated with slower fibrosis progression, both of which were located in the extracellular domain of the TLR4. And just to remind you, there are 2 different intracellular signaling pathways. We focused on the MYD88 pathway, and we used an approach that's illustrated in this slide to examine how those SNPs might actually change fibrosis progression rate. We did a very simple, but in the end, a very informative approach, and this was work of Jinsheng Guo that was just published. We isolated stellate cells from TLR4 null or MYD88 null mice, which are viable, and we reconstituted TLR4 expression using TLR4 SNPs either with wild type or cDNAs with either wild type or SNP sequences, and then simply assessed classical responses downstream of TLR4, in this case NF-kappa B, and a whole range of cytokine and fibrogenic gene expression patterns as well as apoptosis. I'm only going to show you one bit of that data just to illustrate at least how we've begun to link the biology. And what you're looking at here is MCP1 protein expression. This is an inflammatory chemokine that's a well-known downstream consequence of TLR4 signaling. In wild-type stellate, and these are stellate cells, again, either wild-type or null cells reconstituted with either the wild-type human cDNA or the different SNPs, or in this case, MYD88 null cells. And what you can see is there's a gradient of decreasing expression or secretion of MCP1 in cells expressing either the single or the dual SNPs associated with slower fibrosis progression. Now, admittedly, this is a simplistic approach. Clearly, TLR4 is expressed in a lot of different cells. Ultimately, we need to interrogate each one of those cells to see how the SNP alters the biology and might also explain the altered fibrogenic response. But at least as a first approximation, it begins to give us a link between the pathology of liver, the rate of fibrosis progression, and the biology of stellate cells vis-a-vis these SNP sequences. We've subsequently gone on to look at most of the other SNPs in that list, and I don't have time to go through that today. So let me come back to the issue of regulation of fibrosis and introduce an approach that we took some 10 years ago that has turned out to be very interesting and full of surprises. As we assembled this fairly complex and what we viewed as a comprehensive understanding of the phenotypic changes of stellate cell activation, we began to ask what drives this response or this cluster of responses, and we did something that probably nobody under the age of 40 will ever do again, which is subtraction cloning using old-fashioned biochemical approaches in which we were simply asking what are the genes that are induced as the stellate cell activates, acknowledging that part of the activation could also represent a downregulation of some genes that preserve the quiescent state. But in this case, we were really interested in induced genes. And among several that we cloned, one in particular is illustrated here. It's known as KLF6. This is actually a member of a growing family of molecules of Kruppel-like factors that have a growing range of activities. Many of you may know the most famous now, KLF4, because this represents one of the critical factors that Yamanaka and his colleagues described as conferring pluripotency in the generation of induced pluripotent stem cells. But back to the KLF. So all of the KLFs have a virtually identical DNA binding domain with these 3 zinc fingers bound together in a tetrahedral complex. By zinc is C2H2 zinc fingers, and each of the Kruppel-like factors has a unique N-terminal activation domain. So it became clear to us that KLF6 in fact was ubiquitous. It was expressed in all cells. In addition to our interest in stellate cells, we wanted to understand how it contributes to hepatic regeneration and growth responses. And so we did basically RNase protection and looked at the change in mRNA after partial hepatectomy in hepatocytes. And what you can see is just as in stellate cells, which I haven't shown you, it's an immediate early gene. It's very rapidly induced and then tapers off. We wanted to understand what it did in vivo. Ideally, we wanted to generate a mouse that expressed this in the cell we originally cloned it from, stellate cells, but at the time there were no stellate cell-specific promoters, and so knowing from this data shown here that it was also regulated in hepatocytes, we chose to use a much more conventional hepatocyte transgenic approach with a promoter to the transthyretin gene, which drives hepatocyte-specific expression of a transgene. And shown here, surprisingly, were the KLF6 hepatocyte-specific transgenic mice. They were runted, they had small livers and reduced cell growth, which was quite a surprise since this was a gene that was induced during stellate cell activation, presumably a growth stimulatory state. And sometimes it's better to be lucky than smart. I happen to have just come back from a year sabbatical as a Fulbright scholar in the lab of Moshe Oren, Arnie's former postdoc and well known to many of you. So I was thinking a lot about tumor suppressor mechanisms, even though I really hadn't had any interest going into it apart from just using P53 as a great model system. And lo and behold, in the transgenic mice, there was increased expression of P21. We went on to show that, in fact, this is a P53-independent transactivator of P21, and it was associated in those isolated cells with reduced PCNA expression. So this really started us on a completely unexpected pathway of saying, well, maybe this is actually a growth suppressor gene. And more importantly, since we had originally mapped it to chromosome 10p15, a region that was lost in a number of tumors, maybe in fact it was a tumor suppressor gene. So we began looking at a number of different tumors. I'm going to show you mostly, but not exclusively, data in hepatocellular carcinoma, and I'll summarize some of the other data. What became clear is in a panel of hepatocellular carcinomas that in general the mRNA level compared to normal was reduced and sometimes significantly reduced, which would at least be consistent with the loss or inactivation of a growth suppressive transcript. It turns out that because we have a very large, very active liver transplant and liver resection program at Mount Sinai, we also have the benefit not only of collecting samples, but of also following patients prospectively to look at the impact of different genetic changes on their outcome years later. And one of those analyses is shown here. So it turns out that those patients whose tumors had very low expression of KLF6 mRNA, that is less than 10% of normal, which in this case was 13 patients in our original cohort, had a markedly reduced survival. Whereas those who had greater than 10% of normal, they certainly had a reduced survival owing to the presence of a tumor originally, but their prognosis was significantly better. So we generated an antibody, and we expected to see reduced expression in a number of different tumors, and this was where we got yet another big surprise. We started looking at colorectal cancer, and what you're looking at here is KLF6 staining of a paired normal and normal colon and an associated tumor in the same patient. Here again, normal colon and an associated tumor, and what's pretty clear is there's anything other than low expression of KLF6. In fact, the expression is markedly increased, and what you can see on this high-power blow-up from this field is that this is largely cytoplasmic. And so this was puzzling, and ultimately it led to a discovery that's summarized in this slide, which is in fact that KLF6 is alternatively spliced. The original data we generated came from prostate cancer, and I'll emphasize this was a collaborative and still is a collaborative activity with John Martignetti and Gautam Narla. Gautam originally was my MD PhD student. Now he has a lab of his own at Mount Sinai. And when we originally looked at just simply flanking PCR using 3' and 5' untranslated region primers, we got a number of bands, and that's how ultimately we discovered that this is alternatively spliced. And in the case of prostate, what looked interesting, even though this was not quantitative, is as the tissue progressed from normal to localized and ultimately to metastatic prostate cancer, the predominance of these lower molecular weight isoforms that were ultimately cloned and sequenced and illustrated here began to increase. And in fact, in a paper published last year in JCI in collaboration with investigators at Sloan Kettering, it's very clear that metastatic prostate tumors that have higher levels of expression of this splice form, which we call KLF6 SV1, have a markedly poorer prognosis. Interestingly also was the fact that in the splice variant 1, there was a frame shift that generates a 21 amino acid sequence that's not present in the wild type. We still don't know the function of this N-terminal sequence, but obviously we're quite interested in also generating monoclonal antibodies to understand what the biology is that's conferred by the generation of this alternative transcript. Furthermore, it became clear that the reason we were seeing intense expression in the cytoplasm of some tumors was based on what this in vitro study in 293 T cells. When we transfected flag-tagged SV1, SV2, or wild type, it was clear that whereas wild type KLF6 was largely nuclear, although not exclusively, both of these isoforms, which on mapping lose a nuclear localization signal here, actually are largely cytoplasmic. And we think it's because they lack an NLS. We also know that they have an increased half-life, and although we don't know the exact mechanism of what's emerging as a dominant negative activity, we do know that it can heterodimerize with the full-length KLF6, and our working theory is that this sequesters the full-length KLF6 in the cytoplasm. So we've gone on to characterize these kinds of defects, that is, not only loss of heterozygosity, which I didn't review today, or inactivating mutation, but also whether or not there's alternative splicing in a large panel of tumors. And these fall out into really two general patterns. The first is shown here, which fortunately were the first that we looked at. That is that there's a high rate of LOH and mutation in prostate, colon, and somewhat less so in hepatocellular carcinoma. In the second cluster of tumors, there's virtually no mutation, modest to marked amounts of LOH, but virtually in every tumor we've looked at, there's increased alternative splicing of KLF6. I don't have time to go into all the mechanisms, but let me just show you a little bit of what we've learned. Certainly, we went back to the tumor of interest, that was the hepatocellular carcinoma, and to our pleasant, I wouldn't say surprise, but as we would have hoped, as the tumor advances from early to advanced and then very advanced, the relative expression of this splice form SV1 compared to the full-length KLF6 increases. And going back to our natural history data, it turns out that the tumors that have increased SV1 relative to full length with a ratio of greater than 4 have a markedly increased recurrence. So in other words, this is without recurrence, so as you go down more rapidly, you're recurring more quickly, or the tumors are recurring more quickly. So the paradigm that seems to be emerging is that whereas KLF6 SV1 may affect survival, the recurrence, the metastasis, if you will, if this is what these represent in liver, metastasis is driven by SV1 generation and alternative splicing, which we tend to think is a relatively later event in the pathogenic spectrum. So not surprisingly, we were interested in the impact of allelic loss on carcinogenesis in an animal model. We tried to generate null mice. The null mice are embryonic lethal. Interestingly, KLF6 also contributes to hematopoiesis and endothelial development through what we think is the generation of the hemangioblast. So at this point, we've just generated floxed KLF6 mice with liver-specific deletion, have an extremely interesting phenotype that I don't have time to review. But what I can show you is that even the heterozygote mice are more tumor-prone when stressed with a carcinogen, in this case, DEN or diethylnitrosamine. So what you're seeing here is simply the fact that even at early stages, and this was 20 weeks, animals that get a single dose of DEN at day 15 have increased tissue disruption, a lot more steatosis and disruption of the lobule. And then, much to our delight, they also have a markedly increased risk of tumors after DEN. They do not develop spontaneous tumors with loss of a single allele, however. And so what you're looking at here is both the total number of macroscopic lesions as well as the mean diameter of those lesions in the KLF6 het mice compared to KLF6 wild-type mice that also received a single dose of DEN. And then finally, we are interested in the therapeutic impact of antagonizing the alternative splice form that appears to be oncogenic, and I'll just show you a little bit of data that's been published some time ago, looking both at colony formation assay and also using tumor xenografts. So in this case, what we're looking at is colony formation when we, in PC3 cells, when we first treat them stably with an SI to SV1, and what you can see is there's a fold decrease in both colony formations and then in large colony in particular. And then finally, we think that this has some potential therapeutic import. This is one bit of data. We have actually more compelling data by my collaborators in ovarian cancer and other tumors. But what we did here is to use tumor xenografts where we either stably expressed SI to luciferase to the full-length KLF6, which predictably would increase growth, as it does here in these isolated tumors and in the aggregate data here, or SI to the SV1, thereby inactivating an oncogenic form of this gene. And so what you can see here is the tumors are smaller, and that's just simply shown by the tumor mass here in tumors that were injected into nude mice that were stably expressing SI-SV1. And what you can see here is the SV1 knockdown is confirmed in the tumors at the time of harvest. So we're very interested in using this SI as a therapeutic tool, particularly in tumors that have high expression of the SV1, because among other things, it looks like SV1 confers resistance to apoptotic stimuli. So we think that this should be a sensitizer, if you will, to conventional chemotherapies that induce apoptosis, for example, like Taxol. So the next question is, if there's increased alternative splicing in cancer, what's driving it? I'll show you a little bit of work from Steve Ye, who just is graduating next month with an MD PhD and going on to pathology at UCLA, and this is Steve's thesis work. And simply what he did is screen with a whole bunch of different cytokines and stimuli to see if any of them selectively affected splicing, predominantly focusing on different hepatoma lines as well as some colon cancer lines, which I'll show you in a minute. And what he found is that TPA increased the relative expression of the splice variant 1 in all 3 cell lines. And knowing that this, among other things, led to RAS activation, he went down the path of looking for RAS activation as a particular determinant of KLF6 splicing. And you can see here in the benign prostate cancer line, BPH1 stable expression of a KRAS V12, increased splicing. And interestingly enough, it had no impact on PC3M, which is a highly metastatic line, because these cells already have very, very high levels of KLF6 SV1 expression. So we presume that unlike this cell type where there's modest expression, here there's already marked expression of SV1, and so the impact of RAS is much less. Moreover, when we looked at human tumors, there was a statistically significant correlation such that tumors that had low alternative splicing had low HRAS expression. In contrast, tumors that had high HRAS expression had high alternative splicing. And we're going on now to develop animal models to see how we can manipulate this not only in tumor models but also in growth conditions. Some other ways of approaching this was to use a farnesyl transferase inhibitor, in this case FTI, showing that there was selective decrease in the SV1 variant 1 in all 3 cell lines. And then finally, we obtained cell lines from investigators in England that were derivatives of the HCT116 colon cancer cell line that were lacking a single allele. And what you can see here is where there was no impact of allelic loss on the relative expression of KLF6 full length. There was a progressive decrease in these 2 sublines or daughter lines, each of which lacks 1 allele of RAS. So that the dosage of RAS seems to correlate with the amount of alternative splicing. And then finally, to begin to understand what some of the pathways might be that are regulating the alternative splicing in terms of the splice machinery, which we're only learning about now, Steve used a number of different SI approaches to well-characterize splice factors, and the one that seemed to yield the highest increase in splicing was when he knocked down the factor known as ASF-SF2. And that was again true in all 3 cell lines where there was an increased relative expression of SV1, and that led to a model that's shown here, in which ordinarily in the absence of RAS signaling, ASF-SF1 directs most of the KLF6 transcript into a full-length isoform, yielding transactivation of P21 and growth inhibition. And through pathways and other factors that we don't completely understand, we think that RAS activation ultimately uncouples ASF-SF2 from the transcript, and in some way that we still don't understand, yields a predominance of the KLF6 SV1, which of course has a dominant negative effect, thereby abrogating P21 and promoting cell growth. So just to summarize this component, and then I just have a couple more points I'd like to make. What we can say now is that KLF6 mRNA is reduced in most hepatocellular carcinomas. It's inactivated through a variety of pathways or mechanisms: loss of heterozygosity, inactivating mutation, and splice form overexpression. But interestingly, the reduction in the full length is associated with reduced survival, where the reduction, or rather the increase in splice form, is associated with increased recurrence, i.e. increased metastasis. And this is a pattern we've now seen in prostate and lung and in ovarian cancer as well. We've also identified very high rates of splicing in glioblastoma. So this raises a number of questions, and I'll just tease you with some data that's just beginning to emerge. What we've also found is that the prevalence of splicing is actually much higher in tumors derived from hepatitis C than hepatitis B. And we've begun to work with Charlie Rice, who's among the pioneers in developing cell-based infectious models for HCV. And it looks like HCV in some way may actually directly increase KLF6 splicing using a mini-gene reporter construct that we introduce into his cells. And this is something that hopefully I'll have an opportunity to talk about to you sometime in the future. And it looks like it's HCV specific. Certainly we'd like to know what the implications are of RAS activation in vivo, with the obvious likelihood that, or the presumed likelihood that as RAS activation occurs, one of the downstream consequences of this later on in tumor development is increased alternative splicing. And then certainly we're not naive to the prospect of using SI if the SI technologies continue to mature, so that we can antagonize SV1 as a therapeutic approach to sensitize tumors to more conventional chemotherapies. So finally, this brings us back to the original problem, which is how do stellate cells activate, and why on earth did we end up cloning a tumor suppressor in a context in which we were looking for genes that turned on this mesenchymal cell? And you can probably guess at the answer. So it turns out that what we found, so let me just state the paradox again. KLF6 is induced following liver injury when stellate cells proliferate, but KLF6 is growth inhibitory. And what it turns out, we had originally cloned in retrospect, was a region of the 3' untranslated region that ultimately was part of KLF6 SV1. And because we used that fragment to screen a full-length library, we effectively bypassed, because we weren't interested in partial transcripts. So we favored the cloning of the full-length transcript by using a fragment that initially probably represented one of the splice forms that's overexpressed. And that leads to a working model that's just shown here in this final slide, which is that our working model in a non-cancer context is that we think liver injury and oxidant stress associated with increased RAS, which has been documented in stellate cell activation, ultimately leads to a change in the ratio of KLF6 splice form to full length. So now we have a predominance of a growth-promoting isoform. And ultimately, this leads to further activation of stellate cells, increase in a number of transcripts associated with that phenotype, and phenotypic markers in the tissue that are associated with progressive fibrosis. And so we're testing this now using a number of different animal models where we finally have stellate cell-specific promoters to drive splice form expression in vivo in liver. So let me just summarize what I've tried to introduce today. That hepatocellular carcinoma is rapidly increasing as a neoplasm associated with advanced fibrosis. KLF6 cloned from activated stellate cells turns out, to our surprise, to be a tumor suppressor gene. And then finally, we think that alternative splicing of KLF6 has increased. Well, we know it's increased in many cancers. It's regulated by RAS, at least in models. It's associated with RAS activation in hepatocellular carcinoma. And we're equally interested in how this relatively unregulated response in cancer, in tissues that are responding to injury in a more regulated fashion, this splicing may also be regulated. That is, not only what turns it on, but what turns it off. And so there's clearly a lot of areas we're pursuing, and as I said, hopefully there'll be more data to come in subsequent years. Clearly, having 6 different job descriptions prevents me from working at the bench virtually at all, and the people that have are shown here. I'm going to emphasize already Jinsheng Guo's role in the genetics studies, Steve Ye in the RAS studies, Sigal in the hepatocellular carcinoma, and Helen for the colorectal cancer. And then I've been blessed with great collaborations, both by John Martignetti and my former student Gautam Narla, Josep Lovet, who's part-time in our institution and in Barcelona. Barcelona is probably the leading authority on hepatocellular carcinoma clinical trials, and he's responsible for amassing this extraordinary tissue bank with outcomes data that's been so informative for us. And then finally, for the genetic studies, John Sininsky of Solera. So again, I want to thank Arnie very, very much for including me in this symposium, and hopefully I have time for a few questions. Thank you.
Questions? Bob?
I wanted to ask, in the prostate experiment, when you looked at the siRNA experiments, were they only in PC3 cells, or were there other cell lines used for prostate by chance?
The published work was only in PC3, and I think Gautam has done additional studies that is in LAN cells, same effect. PC3 are particularly appealing because their endogenous SV1 levels are extremely high. And as I also underscored, this has already been sort of tracked with outcomes in human prostate cancers. So those prostate cancers that have the highest SV1 are the most metastatic and have the poorest outcome.
Can I also ask, was there dependence on AKT, did you say? AKT status?
We don't know yet.
Okay.
We don't know. We speculated in terms of the RAS, but we don't know.
Why is it in liver cancer the injury is responded to by excessive fibrosis? Why is that a good thing for the liver, or why did it do it?
Well, I guess the question is, what is the organism trying to do? And I suppose the real answer is, when confronted with an insult, the first thing to do is encapsulate it with scar. From an evolutionary perspective, if you can keep the liver, the injury constrained and the host of that tumor or that injury fertile long enough to pass on their germlines, that's probably all you need. And the downstream consequences of fibrosis 30 years later and carcinoma are of no consequence to evolution. So I think what the liver is really doing is simply trying to encapsulate a region of injury. It's not the only organ where fibrosis is a precursor to cancer. Probably even more dramatically, pancreatic carcinoma is typically highly desmoplastic, and so and pulmonary to some extent, but not nearly as dramatically as liver. And again, liver is really the only organ that has this smoldering injury for literally decades before these kinds of clinical consequences ensue.
Okay, there's no further questions. Thank you. Now we're going to participate